I think that there is nothing wrong with having proteins and, of course, building muscle, but the amount that you need to get there is much lower than what people think. Don't forget, though, acute versus 110 healthy, right? Two different worlds. I can inject you insulin and I can show you lots of benefits and I can publish 1,000 papers. It doesn't remove the insulin resistance later on and the diabetes that you're going to get eventually. So our job is to make people live to 110. Somebody's job is to prepare somebody for the Olympics. That's a different job.
Welcome to the Proof Podcast, a space for science-based conversation exploring the health and longevity benefits that come with mastering nutrition, physical exercise, mindfulness, recovery, sleep, and alignment. Facts, nuance, and trustworthy recommendations, minus the hyperbole. Dr. Longo, welcome to the show. It's great to be able to connect again.
Thanks for having me.
Been very excited to get you back on. We were just chatting, I believe it was 4 or 5 years ago that we caught up in LA and did our first episode together, which was a huge hit. And over the years, I've had many requests to get you back on. And I know that you've done a lot of research since then, and so there's plenty to explore. And over the past month or so, just to give you a bit of context, I've had a bunch of conversations with the likes of Stuart Phillips, Christopher Gardner, Don Layman, Sachin Panda, and plenty of others. And it's become clear to me that while there's a great deal of agreement on certain areas of nutrition, health span, and longevity, there's also some specific areas where folks within the scientific community have formed slightly different positions. And I think one notable area in this conversation is protein and specific amino acids and whether or not higher intakes, which seem to be beneficial, at least on paper, for certain outcomes like strength and bone mineral density, whether this high protein intake is deleterious or not when it comes to the development of diseases such as cancer and cardiovascular disease and our lifespan, things that you talk about a lot. So this is really what I want to focus this episode on with you today, zooming in on things like IGF1 and mTOR, kind of buzzwords that I feel a lot of people are talking about, but I'm not sure if everyone fully appreciates what they mean. Protein source, protein amount, cancer, and then if we get time, some of your research looking at fasting as a sort of adjunct therapy during immunotherapy for people with certain forms of cancer. Why don't we kind of start here with your sort of very high-level position on nutrition, protein disease, and longevity, and sort of where you've landed based on all of your research that you've done over decades now.
Yeah, so I wrote an article recently with Rosalind Anderson of University of Wisconsin in Cell for the scientific journal Cell. And those that are, technically oriented, let's say, can get a copy. It's free. You can just download it. And the title is about nutrition for longevity. So, what are all the complexities for someone to make it to 100 or 110? But more so, what are the complexity to make it there healthy and relatively strong, right? So, what emerges is a fairly complex type of nutrition that has, of course, intervenes on the IGF1 and the growth factors and all of that, but also is done in a way that does not lead to frailty. It does not require, for example, calorie restriction. Calorie restriction refers to this famous, very old intervention, very simple, which is what happens if you take a person and you reduce the calories by 25% and you don't change the dietary composition. And what happens, at least in the monkeys, the monkeys do extremely well. And they have very low cholesterol, very low blood pressure, et cetera, et cetera. But then the monkeys are pushed to a limit, and you could argue over the limit. And they look very frail. So they're very thin. The muscle mass is very low. They could be not necessarily the muscle strength could be relatively good, but they're pushed to the edge. And then the monkeys don't live them as long as you would think, right? So yes, they have all these incredible markers or levels of these markers, but then they just live a little bit longer than those that had the exactly same diet but 25% more calories. So then I think that if you look at everything, you come up with pescatarian diet, nutrition, up to say between age 20 and 65. I think that should change between 0 and 20. I wrote a book in Italy, which hopefully soon will be translated into English. And so probably the Mediterranean diet seems to be best for the 0 to 20 and the 70 to maybe 90 range. But in between 20 to 70, let's say what I call the longevity diet, I think it's a better choice, and it's a pescatarian diet, low protein, high nourishment, high moderate levels of unsaturated fats, the olive oil, the nuts, etc., and then whole grains, and completely avoiding meats, red meat and white meat. Now, some people might think of this as extreme, but in fact, as you know, I talk about 5 pillars. So not too much of this is my opinion, right? I mean, of course, I put it together. That's my job, and we do the research that is missing. But if you put it together, so if you think about basic research, like how do you make a mouse live longer? If you think about, well, what happens if you put 2 groups of people, randomized clinical trials, if you have diet A or diet versus diet B, then what happens in epidemiology? What if you look at millions and millions of people and eating different patterns? Now, to a lot of people, the epidemiology is the only pillar, right? And that's where I think a lot of this controversy comes in, right? So a lot of my colleagues, unfortunately, and I mean, not all of my colleagues, but certainly a lot of colleagues in the food world, they rely heavily on epidemiology. And I've done a number of epidemiological studies myself, and I can tell you that in epidemiology, it's an incredible pillar, but it's also, you can get a lot of answers, right? You can get anything in the opposite based on how you select the population that you choose to compare. Then also the centenarians. What about centenarians? What about people that have made it to 100 or 110 or 115? Is there something is there a common denominator between all of these that we feel it's hard to argue with? And that's what we've been focusing on.
Okay. You mentioned there that you've kind of got this framework where the amount of protein that might be sort of, say optimal for someone at different stages of their life may vary. And you spoke about kind of low protein in adulthood up to 65, 70. I know you wrote a paper on this with Morgan Levine, which I want to come to. I have some questions. And then once you get to 65, 70, there might be some benefit in slightly increasing that protein. Based on my understanding and reading of your work and hearing you speak, much of that comes back to, or it seems to, these sort of growth pathways of mTOR and IGF1, particularly this idea of sort of through at least the midlife having a low protein consumption. And if someone's hearing this for the first time, these keywords of mTOR and IGF1, and let's say you meet someone at a dinner party, Valter, and they say, Dr. Longo, you're speaking another language. Slow down. What do these abbreviations actually mean? And for the sort of lay person out there, where are these pathways found in the body and why are they important? What is their purpose?
Yeah, so IGF-1 refers to insulin-like growth factor 1. And so, not surprisingly, is the main factor in the blood making us grow, right? So we follow these little people in Ecuador and they have extremely low IGF-1, and they're about 3 feet tall usually, right? So they're born with this growth hormone receptor deficiency, and the result of that, so growth hormone basically regulates the levels of insulin like growth factor 1. So if you have a low growth hormone receptor, you have low IGF-1. And the other one that you mentioned is mTOR, target of rapamycin. And so this is a X downstream, but also somewhat parallel to IGF-1. And so mTOR is also at the center of growth. And this is how we identified the mTOR cis kinase pathway over 20 years ago. Its role in aging, because we saw that when we mutated very simple organisms, the ones that lived the longest were dwarf. They became much smaller, but we could make it live 3-5-fold longer than the regular ones. And that was a mutation in the mTOR cis kinase pathway, meaning we inactivated mTOR cis kinase, and all of a sudden these little microorganisms, they could live a lot longer. So yeah, that's a target of rapamycin. Rapamycin then, not surprisingly, is a drug that ended up being demonstrated to extend the lifespan of mice, and in a way that I think most people will say is superior than anything else we've seen in history, right? So there seems like blocking rapamycin, especially if you start in middle age, works better than anything we've ever seen. So that those are mTOR and IGF-1. And so we and many other, in fact, I almost 10 years ago, I organized a conference where we brought all the top geneticists and experts in the biology of aging from all over the world. And we asked the question, what is the best target to extend human lifespan and health span? And the consensus was that the growth hormone IGF-1 was the very top. It's the one that got most votes. It didn't get everybody's vote, but certainly got most votes. And in that paper, we wrote a paper about that. And then in the paper, we actually put the votes. We wrote down who was present. All the present were authors of the paper. And then so then I think that not the consensus, but certainly the most votes go to growth hormone receptor or IGF-1.
Are these pathways in all tissues of the body, or are they only found in certain cells like muscle cells, or will you find sort of mTOR and these kind of growth pathways throughout all tissues?
mTOR is everywhere, and so is I would say the IGF-1 is in the great majority of cells. Probably not all of them, but let's say the great majority. Now they in different cells and different tissues, they can have very, very different effects, right? So some cells in some moments have the job of making glucose, like liver, and some cells have the job of, at the same time, of using glucose, right? So you can imagine how a brain cell and a liver cell during a starvation period would do something very, very different. One is trying to deal with fasting, and one is trying to do its job of functioning normal as possible, right? So yeah, but then just to tell you, these genes and proteins are everywhere, but they can have quite different jobs depending on the state.
Mm-hmm. And I mean, I feel like there's some kind of general fear out there around these pathways, but from my understanding and reading your work and others, is that these are very, very important and they're critical. But are we talking about sort of when they become hyperactive, overactive, that we see deleterious effects in physiology?
Yes, I think a good example is insulin, right? So if you think about 50 years ago and you think of insulin, and you think of insulin as a very, very positive hormone, and then, but then it took a while, right, to realize that if you have too much insulin all the time, then you become insulin resistant. and then you have diabetes, right? So it's the same or similar with these growth factors that you have to have the right amount, and there's probably at some point in some tissues you might develop resistance to these growth factors, but you also might develop hyperactivity of these factors, leading to all kinds of problems. And so in cancer, certainly lots of epidemiological studies and our own studies and mouse studies, and so there's a lot of different type of studies, including the genetics one, suggesting there is a tight link between the levels of IGF-1 and cancer, but also the levels of IGF-1 and lots of other problems. So meaning that if you have a lot of it all the time, you're probably not going to do very well. And not surprisingly, people that have acromegaly, so they're born with very high levels of IGF-1, they have a short lifespan, and they die fairly early from all kinds of problems.
Mm-hmm. I want to come back to a couple of things there, but just to kind of continue to kind of frame everything that we're discussing here. If someone's thinking, well, what causes IGF-1 to get elevated? My understanding is that it's the pituitary gland produces growth hormone, which then stimulates the liver to produce IGF-1, or a lot of it anyway. Is that where most of IGF-1 comes from in our body, or is it also influenced by hormones that are in our food?
No. Most of the IGF-1 comes from the liver and the circulating. Now, IGF-1, lots of cells in the body can respond to growth hormone, or even without growth hormone, and generate IGF-1. But the circulating IGF-1, the great majority comes from the liver in response to growth hormone from the pituitary, making it to the liver and causing its release.
Mm-hmm. And so what influences how much growth hormone we produce?
Well, proteins, not surprisingly, are the influence number one. So if you take a group of people, as it has been done, so if you take a group of people and you put them on a low protein diet for long enough, you're going to see the IGF-1 in response to different cycles or cycling of growth hormone. You're going to see IGF-1 go down. And also, if you do fasting, like in our clinical trials, many clinical trials now, if you do fasting by day 3 or 4 of fasting, you'll see IGF-1 go down. And I think that it was there was a study where they did that, and then they just fed proteins, and that was enough to bring the IGF-1 back up.
Yeah, the IGF-1 levels are typically lower in people following sort of plant-based dietary patterns like pescatarian diets and vegetarian diets, right?
Not necessarily. There have been studies, lots of vegetarians and even vegans, they know that, at least the ones that are worried about their health, they know that the levels of certain amino acids and proteins are low in their diet, and so they overeat. They overeat proteins, and so a lot of the vegans and vegetarians have normal IGF-1 because they have so much proteins. Now, that doesn't mean all of them. Some of them are probably very much IGF-1 deficient. But in studies that have been carried out, the vegans had not a statistically significant difference in IGF-1 compared to the controls, unless they also were protein restricted. Because being aware of the problems with proteins, I think they were over 20% of their calories came from proteins.
Yeah, which brings me to, you mentioned before sort of chronic elevations being a problem. So how do we kind of differentiate between acute changes in IGF-1, say after you do resistance training or have a one high protein meal versus chronic elevations? And I guess if we were to think about this with a different biomarker, perhaps we could think about blood pressure. If you do a workout, your blood pressure can go up acutely. If we were just to look at that in isolation, we might say that exercise might not be that great for our cardiovascular health, but we know long term it has a different effect on blood pressure, or typically does. How do we kind of think about that here with IGF-1?
Yes. Well, first of all, in the studies that looked at muscle synthesis, the protein level needed to optimize it was not that high, right? It was around 30, 35 grams from what I remember. And, this was per meal. So you could argue that if you went twice as much and you did 2 workouts with 2 meals, you could get to 60. But those studies, and it wasn't just one, suggested that 60 grams, let's say, of good quality proteins having the right amino acid profile. That study suggested 30 grams, but let's say even go twice as much. That would still be fairly low protein level, right, for most people. And so I think that that there is nothing wrong with having proteins and of course building muscle, but the amount that you need to get there is much lower than what people think. And so our recommendation, we have 2 foundation clinics, one in Los Angeles and one in Milan. And our recommendation is to establish what type of muscle mass and get somebody to measure it. What muscle mass are you happy with? And then, go down in proteins until you actually start losing that, right? So go to don't go below, let's say, 0.7 grams per kilogram of body weight per day. But, as long as you stay above 0.7 and your muscle is fine, then you're good to go, And you could also argue that you're now, IGF-1 and insulin actually are very similar, have very similar receptors, right? So it's possible, it's not very well established, but it's possible that you could be doing the opposite. You could be maintaining the IGF-1 receptor more sensitive, right? By keeping it lower, like you keep your insulin receptor more sensitive by keeping insulin low all the time. And then, of course, insulin goes up with a meal, and now the insulin receptors are very sensitive. And that's what you want, right? You want to have low levels of insulin, but then spike it, have insulin do its job, and then get out of the way. And so probably, although this is by far from being established for IGF-1, but probably you have the same. You can have the same argument for IGF-1.
Okay, I have a bunch of questions, and I think you're right. I think that there are a lot of people in the plant-based community that are aware of differences in amino acid sort of ratios in plant foods, and then just finding specific ways to kind of make up for that. And I'll tell you, I think certainly in the sort of muscle hypertrophy strength community, there's a lot of talk of research that shows sort of optimal protein intake for hypertrophy being at 1.6 grams per kilogram. And that's a threshold, Valter, that everyone is kind of referencing out there, including a lot of professors and science folks.
Yeah, okay. Don't forget, though, acute versus 110 healthy, right? 2 different worlds. I can inject you insulin, and I can show you lots of benefits, and I can publish 1,000 papers. It doesn't remove the insulin resistance later on and the diabetes that you're going to get eventually. Right? So our job is to make people live to 110. Somebody's job is to prepare somebody for the Olympics. That's a different job. Yeah.
Right. So we might be talking about 2 different goals here, and that was kind of my question with regards to let's take me for example. I only eat plant protein. That's where I get all of my protein comes from plants. And I want to come to your paper with Morgan Levine, because I know in there you looked at the effect on cancer incidence. You went a level deeper and looked at protein source, animal versus plant, and you saw some interesting things in there. But let's take me for example. I'm wondering, can I have my cake and eat it too here? So I'm getting all of my protein from plants, but I would be consuming above the RDA, right? And I also fall into that kind of midlife bracket that you're talking about, where low protein you recommend. Do you think, for me, someone who is metabolically healthy, consuming all protein from plant sources, that consuming above the kind of RDA is going to have some sort of long-term deleterious effect on my health?
The data suggests yes, right? So for cancer, when we went from animal to plant-based, those that had more than 20% of calories coming from proteins. If it was animal-based, there was a 400% increase in the risk of developing cancer. And if it was more plant-based, it was a 300%. Now the effect on overall mortality went away when it was a plant. It was we were looking at subjects that were on a more plant-based source. So so then there can definitely be differences, but I think that at least the data would suggest that if it doesn't really matter if it's plant-based or animal-based, once you get so high that the plant-based amino acid profile is equivalent to that of the animal-based, right? So this is probably why the IGF-1, in the papers that were published, the IGF-1 was returned to normal levels from eating more plant-based proteins.
Yeah, that's very interesting. Do you think that some of the benefits that we see in the research, where we see people consuming more plant protein, is also driven by the entire package, the differences in saturated fat and fiber, etc.?
Yes. So the IGF-1 and lots of markers are not going to, TOR, including TOR, are not just going to be affected by proteins and amino acids. Probably the levels of sugar, insulin, and lots of other factors coming from nutrition, and maybe not just nutrition, are going to be playing a role. So so then everything matters, but certainly if you look at the studies, the proteins and the amino acids are playing the key role. Methionine certainly being one of them, leucine being another one. So those leucines certainly seems to be associated with levels of TOR, and methionine and cysteine with the levels of IGF-1. So not surprisingly, plant-based protein sources have low levels of these, so those are at the center of growth pathways, but everything else probably matters. And we also knew this from very basic studies in yeast, where we had the amino acids and TOR signaling goes up. But then if you had sugar, that also drives TOR signaling. So, I mean, this is a very simple system, so we can tell exactly what's happening. As you go to humans, it's very complex environment. It's a lot harder, but probably that's the case too, that multiple components are contributing to growth signaling.
From a kind of mechanism point of view, so you've sort of explained this association that exists: higher IGF-1, higher risk of cancer. And is that because IGF-1 as a growth factor is promoting the growth of precancerous cells, or what's happening at a kind of basic physiology level here?
Yeah, so we actually did this experiment with human blood, believe it or not, right? So the little people of Ecuador have very low IGF-1, and they almost never develop cancer. We've seen one cancer in 50 years out of hundreds of them, right? And this is all over the place. Europe, South America, Middle East. And so and then the mice, they have low IGF-1, they have about 30-40% reduction in cancer, they have delayed cancer, a lot more benign tumor. So it's pretty clear, right, that this happened. And so we took the blood of the little people, IGF-1 deficient, or the relative controls. And then we took human epithelial cells, the type that give rise to breast cancer. And then we damaged the cells with oxidative oxidants, right? So free radicals, right? And so we sort of induce like a cancer-like state or a precancerous state. And then it was very interesting, right? When we added the blood that was very IGF-1 deficient, there were two changes. One we expected, one we didn't think about. One was much lower DNA damage, right? So the blood from the little people of Ecuador, IGF-1 deficient, if the human cells exposed to it were hit with a lot of free radical toxins, the DNA damage was a lot lower compared to the blood, those exposed to the blood from the normal people. And the other thing was the ability to commit suicide was higher. So there's two defense levels. So you protect the DNA, and which we expected from the work on microorganisms, but also we didn't expect so much apoptosis, so suicide was low. The threshold for suicide was lower, meaning that as soon as they get damaged, because the IGF-1 is a very, very well-established anti-apoptotic factor. What does it mean? It means that it prevents the human cells from committing suicide, right? And this is why those pathways, these apoptotic pathways, are very much targeted in cancer therapy, because you want the cancer cell to commit suicide. So as soon as a cell becomes damaged, you want that cell to get out of the way. But if you have high IGF-1, that IGF-1, and now there is probably 10,000 papers on this, right, because of cancer therapy, that IGF-1 blocks that. And then the cell has factors like called BCL2 and et cetera, that are in the same pathways as IGF-1 that block the cellular suicide, programmed cell death.
If we thought of two scenarios here, let's think of, okay, I used myself as an example before, so let's just continue using me here. Protein in my diet coming from plants, but consuming above the RDA, more towards the level that has been shown to kind of optimize hypertrophy, which I appreciate is a very different area of science than what you're interested in here. So as you alluded to, we could be talking about people with very different goals. So it can be hard to compare these directly sometimes. But if we think of two scenarios here, so one is myself consuming that diet and not doing any form of resistance training. And the other is consuming the same diet but doing resistance training, breaking down muscle. And I'm wondering if when you pair it with a lifestyle that has that resistance training and you get that muscle breakdown, do you get the leucine, sort of activating these growth pathways more preferentially in muscle tissue? Or do we have any idea of, with tracer studies or anything like that, of looking at, okay, after you do a workout and you have a very high leucine meal, what percentage of that leucine goes to sort of increasing IGF-1 and activating mTOR in muscle tissue, as opposed to floating around the body and potentially driving growth in precancerous tissues?
Yeah, I don't think you want to transform your audience into biochemists, but I could tell you that we did that, right? So we in a cancer clinical trial in Italy. So we took a lot of women that were on a hormone therapy and cancer therapy, and then we put them on a fasting fasting mimicking diet together with the therapy. But then, because we were worried, this fasting mimicking diet is 5 days of fasting, like let's say nutrition happened every month or so, right? So we were worried about loss of muscle mass. And so what we did was to give them we give them a video where they could do muscle training. And then in between, we give them a Mediterranean diet. I argue, because of what I was saying earlier, I argued that there shouldn't be a Mediterranean diet. It should be my longevity diet, so that we wouldn't help the cancer cells stay alive and grow. The doctors at the oncology hospital argued that they didn't like it's too much. We're gonna give a Mediterranean diet. And what happened was very interesting. Was the women kept gaining muscle mass, right? So of course, in that case, it was a bad idea. But the training, even with the fasting, even with just a Mediterranean diet that was not very high in protein, but sufficient protein. Let's say moderate protein intake. It was not a low protein intake. But with the moderate protein intake, the 20 minutes, I think it's 20 minutes a day of very, very light muscle training. And they kept gaining lean body mass, right? So that my point is that you don't need very much proteins to get the muscle and at the same time get the benefits. And so now our leucine is distributed between muscle and the potential target of cancer. I mean, that's an extremely complicated world that you don't want to think about that. You want to think more about, how do I keep my protein intake and protein profile vegan and low, vegan enough, right? Because then everybody's going to be vegan. But let's say, and this is why I preach pescatarian diet, right? But so how do I keep it vegan enough and low protein enough, but at the same time get as much muscle as I want, right? These women, for example, they probably didn't even want, and that was my argument with the oncologist, right? They didn't want to gain muscle mass. They wanted to stay the same, right? So there was no reason to give them between 1.2 and 1.5 grams per kilogram a day, right? We could have kept it much lower, and I think they will have stayed steady. And in that case, it would have been a much better idea. Now, if you are trying to gain muscle mass, then yeah, you could have gone to, let's say, a fairly plant-based, maybe 1.2 grams. I would say that I seriously doubt somebody does the experiment, and the experiment has already been done, by the way, as I mentioned earlier. So I don't think you're ever going to see any improvements going above 1.2. No matter what, right? So I mean, unless you're really talking about Olympic training, and maybe there is like a very specific super protein loaded period that is going to help you build very quickly. But those studies that I mentioned before suggest that 30 grams per training period, we're optimizing muscle synthesis. Yeah.
Within your sort of overall framework and longevity kind of lens, how important do you think muscle tissue is? Not just from a kind of strength and mobility point of view, but even from a metabolic health point of view, and being such a big sort of sink for glucose.
Not very important. I mean, I think it should be important, but it's not in the sense that if you look at most centenarians that I visited, they look not so good, with muscle, whether they're Okinawans or Italians or in Loma Linda, California. So I don't think that should be the case. I think they would probably be better off because of frailty. So if you look at the Southern Italians, for example, they're very frail compared to the Northern European. They have record longevity, but they're very frail. The frailty is almost twice as high as those of the Northern Europeans. So then the frailty does not seem to interfere with longevity. but it's certainly not a good thing to have because, of course, the quality of life is going to be affected. So I think that, yeah, you probably want to put more emphasis not on increasing protein a lot, but increasing protein a little bit, and for sure increasing strength training, like we did for the clinical trial, so that they could have this, say, the strength of the Northern Europeans and the longevity of these little towns in Southern Europe.
Yeah, I think the exercise and kind of resistance training aspect and trying to do some of that in your lifestyle is something that most people agree on. So just to summarize your position, it's essentially, move your body and try and build a good amount of muscle and strength with as low protein as possible. Would that be a fair statement, at least up to the age of 65?
Yes, yeah, and then and I think that might even help, again, keeping the IGF-1 receptors sensitized, so that when you do have the proteins, everything is working very well. Everything is getting built correctly. Yeah, so they may make, in the long run, in the short run, not maybe 2 grams per kilograms of protein make things easier, but in the long run, I think that the sensitization is going to make things a lot easier. Maybe this is why these women, they probably always had the fairly low, protein intake. They were gaining muscle mass very easily, just doing 20 minutes a day of exercise.
Right. Okay. I hear what you're saying. So potentially, if you're doing a high protein diet to absolutely optimize, say, muscle protein synthesis and hypertrophy today, there could be a trade-off with regards to what your health span is going to look at. So I understand what you're saying there. Can you come back to that paper that you did that found the kind of low protein midlife and high protein later in life? If someone hasn't heard you talk about that, can you just quickly summarize that? And then I have a question about the sort of over 65-year-olds.
Yes, that paper, we basically first look at everybody and say, what happens to people that have a high protein versus people that have a low protein diet, and then and if you look at the entire lifespan, you see absolutely nothing. It looks like doesn't matter. And then if you break it down, 65 and younger, 65 and older, then you see a big effect, right? So the 65 and younger are, if they have a low protein diet, they have a big reduction in the risk of dying of cancer, and also a big reduction in the risk of dying of any cause. But then as the database went to the 70, 80, 90-year-olds, that turned around, and this is probably when you look at everybody, you don't see anything, meaning that the 80-year-old reporting a low protein intake wasn't doing so well at all. And so now that group probably included people that were sick and frail and lots of problems. So that doesn't necessarily mean that an 80-year-old could not have a relatively low protein intake if this person was exercising and was doing training, et cetera, et cetera. But I say overall in the United States, the people that were interviewed, they were not doing very well if they said, I have a very low protein diet, if they were 80 years old. So then then we also did this in mice. And so we took a mouse that was young and we put on a very low protein diet, and nothing happened. And this was like a 4% protein diet. I mean, it was extremely low. And after, I think at least 10 days, but nothing happened. And then we did the same with old mouse. And that old mouse did not like this very low protein diet at all. And within 10 days, it already lost a lot of its weight. So it was probably telling us that, and also from the database, we knew IGF-1 levels. When we looked at the younger people, those that had a high protein diet had a high IGF-1, about 250 or something like that. And then the people that had moderate protein intake had in between IGF-1 level, and those that had the low protein intake had an IGF-1 level that was, around 200 or less than 200. But when we look at the 66 and above group, there was no longer any significant difference between the IGF-1 level. That means that the IGF-1 level after 66 years of age was already low to begin with, and probably having a low protein diet and driving it even lower is not going to help you. It's probably only going to contribute to frailty. So that's part of the explanation of what we see. But
And just quickly, you mentioned so low protein, moderate protein, high protein. Those are kind of relative terms, but low protein was around the RDA, 0.8 grams per kilo.
No, in this case, it was calories. It was less than 10% of calories coming from proteins. Moderate was between 10 and 20, and high was above 20. So if somebody had, let's say, 2,500 calories a day, then people that had more than 500 calories coming from proteins, they were in the high protein group.
Mm-hmm. I think you may have just answered something that I had some difficulty reconciling from this paper. What I think I'm hearing from you is that as you get over the age of 65, protein may have less of an effect on IGF-1. And because I'd sort of read your paper and spoken to people, and one thing that I was left thinking was, if protein increases IGF-1 and elevated IGF-1 increases the risk of cancer, then why would protein all of a sudden be a good thing after the age of 65, given that age is one of the primary risk factors of cancer? So I think you may have answered that, but I'd be interested to kind of hear you elaborate on that.
Yes. This we showed, right? So we showed that IGF-1 does not significantly increase, even between the high protein and the low protein group, right? So not just between the moderate and
over the age of 65.
Over the age of 65, it did not. The high protein intake group and the low protein intake group did not have a significantly different level of IGF-1. But of course, now I had more nourishment, and of course, with the high protein, probably also comes lots of other nutrients. So the story is probably a little bit more complicated, and I think the argument could be that you can still be fairly low protein, fairly low but sufficient protein in the at older age. And that may be even an advantage then, but maybe not a big advantage as it was as it was before. Mm-hmm. So the argument could be that if you kept at the moderate, and that's what we saw, right? The moderate were already doing as well as the high, right? So there was no difference between moderate and high. So it was enough to be in the moderate protein intake range, between 10 and 20% of your calories coming from protein, to get all the benefits. So then that's why we concluded low protein up to age 65 and then moderate after.
Mm-hmm. If someone's listening and thinking, I'd be interested in going and getting my IGF-1 levels tested, is there an accurate test that someone can do, a basic blood test? Do we have kind of reference values that would indicate if someone has a healthy IGF-1 level, if their IGF-1 level is raised and putting them at risk of cancer? And is it something that you would measure in the fasted state, presumably away from meals?
Yeah, so now lots of clinics, almost every clinic in the world can run an IGF-1 test. We published a meta-analysis recently looking at, and basically saying we think IGF-1 levels should be used as predictors of lots of problems, or also, which could also be, extreme frailty. So if you see an IGF-1 of 45, something is wrong, right? So either malnourishment or this is a response to some bad problem. And so then it looks like I think we came up with, from the meta-analysis, about an IGF-1 around 150 or so seemed to be ideal. And so if you look at mortality from all causes, the people that kept 150, they seem to be doing the best. And And then as you went much lower, you saw problems, and if you went much higher, you saw problems. But I think in the 120 to 160, 180 range, that seemed to be that those individuals seem to do very, very well long term.
Yeah, I wanted to ask you about low IGF-1 because I had come across a few papers suggesting that low IGF-1 was associated with greater risk of cognitive decline. I saw another paper that suggested that people with metabolic syndrome often have low IGF-1, which I couldn't quite wrap my head around. But what's going on here? Why is low IGF-1 potentially a problem?
Yeah, and this is why we say, the devil is in the details, and so we already know very, very well that low IGF-1 doesn't cause the problem. Low IGF-1 is the consequence of the problem, right? So the little people of Ecuador, they are born with extremely low IGF-1. They have extremely low IGF-1 their entire life, and they do extremely well. They don't get diabetes often, even when any endocrinologist in the world will say there is no way this person is not going to develop diabetes. So they're protected. And the mice, they have extremely low IGF-1, the growth hormone receptor and growth hormone deficient mice, they have 40% increased lifespan. In fact, I think they still hold the record for lifespan extension, right? So that tells us that IGF-1 does not lower, extremely low IGF-1. If anything, is a solution. Now, keep in mind, this is the circulating IGF-1. It's not the autocrine. So the brain cells and every other cell can still make IGF-1 when needed, right? And so that seems like to be the ideal situation. And so I think it's good to keep very low IGF-1, but if you see low IGF-1 in somebody that goes to the doctor and gets it tested, that tells you there's a problem out there. And this is why with the metabolic syndrome and people that this association that you were talking about is that the body can protect itself. And that's probably what it is, although I don't know that there's enough studies on it, but the body might, because IGF-1 contributes to insulin resistance, maybe the body in response to metabolic syndrome reduces IGF-1 to try to solve its own problem, right? So maybe, and same thing with cancer, IGF-1 can contribute to the survival of cancer cells, and maybe the body, and there were some papers suggesting low IGF-1 in cancer models, and so this could be a response to try to not help the cancer survive. Clearly, and that's what we argue in that paper, that low IGF-1 must be a consequence and not a cause of these problems.
Can you become resistant to the effect of IGF-1?
Yeah, there was a number of papers showing IGF-1 resistance, and I think they were mostly neurobiology papers, but probably many different tissues can become IGF-1 resistant, and so that's understudied. We always wanted to study that. We never quite got around to doing it, but I think it's a very, very interesting. So, for example, muscle cells become IGF-1 resistant, and that's a good question. I don't know if anybody's ever carefully looked at that.
Yeah, well, my kind of brain was immediately, you mentioned before, insulin, another hormone, and as someone progressively becomes more insulin resistant, the pancreas can start to get overworked, right, and it can end up producing less and less insulin. So I was, just thinking that, well, potentially the pituitary gland could become overworked if someone is developing IGF or resistance to IGF-1, which then could see levels go down. But that was just a hypothesis.
That's possible. Yeah, that's possible. And usually the endocrine system works like that, but it doesn't mean in this case that you're going to have the same effects as you see in the pancreas.
Sure. The other sort of interesting area of science that I think muddies the waters here, at least for me, is the Mendelian randomization studies. And I was reading a 2020 paper by Larson, which looked at 400 plus genetic mutations which affect IGF-1 levels in humans. And I believe they did find an association between higher IGF-1 levels and colorectal cancer, but it was quite inconsistent evidence for breast and prostate cancers, and not much evidence for other cancers. What do you kind of make of the genetic evidence that we have looking at different polymorphisms and that affect IGF-1 and rates of cancer?
Yes, I think that I teach my students in my longevity classes not to pay too much attention to any single study, and also to have limited weight on any one discipline. As I mentioned earlier, you have to put it all together, right? So as you put it all together, not every study going to confirm. So somebody that could be genetically predisposed to have high IGF-1 might very well have also developed with the years a second mutation that blocks that negative effect. I'll give you an example. These people in southern Italy are condemned to get Alzheimer, and so they get Alzheimer's in their 40s. And they have a mutation in nicastrin, it's called, and it just condemns them to get Alzheimer's. Then there is some families that have probably developed a second mutation that makes the first mutation not irrelevant, but no longer, so they can now live to, let's say, 90 or 100. So then, if it's genetic IGF-1 regulation, then through the thousands of years or tens of thousands of years, if the first IGF-1, let's say, mutation causes very high cancer rates, it's possible that eventually you select for a second mutation or allele that blocks the first. So that all the families now, they will have both, right? They have a good and a very bad one and a very good one to counterbalance each other. Now, it's just speculation, but I'm just saying, give you a scientific example of why you need the multi-pillar system. And if you look at multi-pillar system, then, the aging research, the cancer research, the epidemiological studies, the ones that I just mentioned, is it possible there's little people with no IGF-1, or the mice with no IGF-1 have record longevity, and now this has been shown by 20 labs, right, including mine. So if you put it all together, if it was a courtroom, I think the jury will say there is no reasonable doubt. That does not mean that everybody with high IGF-1 is going to get cancer, but it just means that you probably don't want to have high IGF-1 all the time, and you want to try to stay in that domain where frailty is low and pro-longevity is very high, and that takes some work, and that's why we have clinics implementing that. Yeah. Non-profit clinic, by the way.
I want to talk about your kind of overall framework and how it works in terms of longevity diet and FMD. Just quickly to tie off, so that blood test you recommended 150. I think that's nanograms per milliliter. Is that the
ml? Yeah, I think it's nanogram per milliliter.
And would that be a test that you would do in the fasting state, like fasting blood glucose?
I think, I think they're always done in fasting, because it's just a regular blood draw, and then they also look at IGF-1. IGF-1 is not as pulsatile as growth hormone, so it's usually pretty steady. It will go down eventually as you stop eating, but not in the morning not in the night fast.
Mm-hmm. Okay. I want to talk a little bit about your research looking at fasting and cancer as a sort of adjunct therapy to immunotherapy. before we slide into that, you've mentioned longevity diet a few times. I think people will be well aware. You're quite well known for the fasting mimicking diet. Just at a kind of high level framework for the average person out there, how does this look? Is this the longevity style diet you're eating on a daily basis, and for the many reasons that we've already discussed, along with resistance training and a lifestyle that helps you reduce frailty? You're doing that daily, and then periodically you're doing a fasting mimicking diet depending on where your health is at.
Yes. So for 30 years now, since my days with Walford, I've been looking for the substitution for the calorie restriction, which I mentioned earlier, right? So, and then I think it took me 20 years, and then about 10 years ago, or maybe a little bit earlier, looking at cancer patients, but also in general, we started asking the question, what if you just, instead of being restricted all the time and being miserable all the time, what if you just, most of the times, eat normally, whatever you can do, whether it's a longevity diet or whatever, not so good diet you may do, and then you just intervene for, let's say, 4 or 5 days. It could be once every 6 months, or all the way to once a month, right, for 5 days. And so now, going forward 10, 15 years, it seems to be working very well. So that, for example, if you think about, I'll just give an example in mice. We took mice and we did a sort of a little bit of a crazy experiment. So what if you just give them high calorie, high fat, and, just the worst diet that you can think of. And then once a month, you give them 5 days of the FMD. And then you put them back in this terrible diet. It's just remarkable. If just 5 days a month of the fasting mimicking diet is able to reverse all the problematic effects of this high fat, high calorie diet. So the cardiovascular disease or conditions, the weight, the lifespan, right? So people on mice on a Western diet live a lot shorter and very sick, cholesterol super high, and they look very much like the human, reflected very much their human profile, and they just 5 days a month completely reverse it. Now, this is not to say keep a bad diet and then do 5 days a month, but that's how powerful that those 5 days could be. And now it gets tricky, right? Because then some people say, well, if 5 days is so powerful, well, why don't I do 30 days, right? Why don't I keep going? Well, the body has a way to then eventually switch you to what's called a thrifty mode, right? So, so you just got to get it right. And in that study, we've shown that the mice, and as we've seen for the people in the clinical trial, they seem to, if anything, accelerate or continue fat catabolism. So they keep breaking fat even after you return them to the bad diet, right? And but what you don't want is the opposite. Eventually, the body enters in this saving mode because it detects a very rough environment, and so then it goes into hypometabolic mode. And this is actually an old New England Journal of Medicine paper. If you push enough too much calorie restriction, the body goes into a saving mode, and now you're burning less fat and less energy, so your energy expenditure is reduced. Now you got a problem, because now the body, the system is gonna want to regain the weight, and maybe even overshoot the regain of the weight and gain more weight than you had originally, right? So this is why we tell people, be careful, because there is a lot of mechanisms, and you need to understand them very well. Otherwise, you're going to have a problem.
Mm-hmm. So what exactly is the fasting mimicking diet? It's 5 days of less calories, specific foods. What does it look like?
Well, the fasting mimicking diet is actually looking at which ingredient, as I mentioned earlier, affects IGF1, which ingredients affects glucose level, insulin levels, and which ingredient makes the person happier, and so society is high, and the hormones that they control, leptin, ghrelin, etc., are in the right place. So it's essentially it's a high plant-based fats, low protein, very low sugar, low calorie diet is, and we have all kinds, right? For cancer patient is about 600 calories a day. For normal people, it's about 1,100 calories a day, day one, and then goes down to 800 calories a day, 2, 3, 4, 5. And the idea is to, if you measure IGF1, IGF-BP1, glucose, ketone bodies, they should be very similar to those that you will obtain if you were just doing water-only fasting. Usually, from lots of work in mice and humans, it's about one. So 5 days of the FMD are equivalent to maybe 4 days of a water-only fast. But the water-only fast, obviously, it's extremely difficult to do, but it's also fairly dangerous, and because of hypotension, hypoglycemia, and lots of other problems. So people could do it, but I would not do it outside of a specialized clinic.
If someone's wanting to do FMD, where's the best place for them to go to learn more about that?
I cannot say it because I'm the founder of a company that does that, and I could donate everything to charity or to research. So I think that unfortunately I cannot talk about companies.
Right. But you can get a copy of your book and you can Google it and probably find it pretty easy. In terms of like how often someone would do FMD, I've heard you speak before. Is that going to depend on where someone's health is at? Say, for example, someone who has really good metabolic health, is healthy body weight, very active, versus someone who has type 2 diabetes, uncontrolled blood glucose, for example.
Yes. So now we're very happy that we in the sense that our collaborators, let's say, have done lots of studies now on diabetes, pre-diabetes, and so it works very well for pre-diabetes and diabetes. And now a new study came out of University of Heidelberg where they did once a month. Both University of Heidelberg and University of Leiden in Germany and Holland did the studies on diabetic patients. Both worked extremely well. And these were monthly cycles of the FMD, right? So monthly cycle of the FMD. But at the end, even in the 12-month-long study, not most people or a lot of people did not do it every month, so they skipped some cycles, but it still was very effective. Let's say if they did it between 6 and 12 times in year one. So if you're a diabetic, probably starting once every month or once every 2 months is going to be the way to go, with the hope that, so for example, we just, at the clinic in Milan, we treated a doctor and he had diabetes. And so on year one, we did a combination of the longevity diet and the fasting-mimicking diet. He did it maybe 5 times in the first year and a half. And then, 2 years later, he was diabetes free, right? So now he maybe does it a couple times a year once he's back in the normal state. So, so then I think that's how you want to think of it. You do it when you need to do it. A diabetic person, maybe once every month, every 2 months. Somebody who's pre-diabetic, maybe every 3 months. And then somebody who's an athlete and extremely healthy and very insulin sensitive, et cetera, maybe, a couple times a year. Now, keep in mind that in mice, we clearly shown this regenerative stem cell-based regenerative power of the fasting-mimicking diet. What does it mean? That whether it was the pancreas, the gut, the nervous system, lots of different systems, the blood system, the hematopoietic stem cells, the fasting-mimicking diet caused the turning on of stem cells. And then during the refeeding period, these stem cells went to work and contributed to the regeneration of various systems. Now, in people, it's harder to prove it. So we're starting to have evidence for that, but it's going to take a while. So then I'm just trying to go away from the diabetes into processes that everybody will want. If this is also true in people now, this fasting-mimicking diet, refeeding cycles can introduce a multi-system regenerative effects that that could make a big difference in lots of organs.
How much do you feel, or do you have a sense of how much of this kind of regeneration and improvement in certain metabolic or biomarkers of metabolic health when doing FMD are driven by calorie restriction and weight loss versus the kind of specific foods and nutrients that are within that diet?
I mean, obviously, you can get lots of the effects of the FMD with water-only fasting. We do have papers where we show that, for example, the prebiotic content of the fasting-mimicking diet was very important in the effects in the inflammatory bowel disease in the mouse model. So I think it's both, but the weight loss in our latest human paper, we're actually showing no correlation with the weight loss, meaning the weight loss happens, it's probably beneficial, but it wasn't that people that lost most weight were benefiting the more on lots of markers and risk factors. So yeah, so there was no correlation with the weight loss, suggesting that the weight loss is not key in this, but it's more the pushing the system to a state, a metabolic switch, right? So if you just think about diabetes, right, and the origin, so we look at diabetes as a disease, and then you give people lots of drugs, and but if you think about 10,000 years ago, people in the summer had to become probably overweight or obese. And I always talk about the emperor penguin, right? The emperor penguin, they become fat because they're going to go about 2 to 3 months with no food at all, right? So every year they become fat and they become insulin resistant. And so they put away the fat that allows them to survive for those 2 or 3 months where they have no food at all. And so this is clearly our history too, right? So we used to have moments of lots of fruits and lots of nuts and lots of honey. So yeah, you had to become fat during that period. And then what unlocked that state was probably fasting, the first 5 days of fasting. Now, probably, if, as I was saying earlier, if you continue, now you go from a fat accumulating mode to a low energy expenditure mode, right? So, yeah, so then that's why the fasting mimicking diet and not the water only fasting. You want to switch person to the insulin sensitive mode, but not to the low energy expenditure mode, right? The true starvation response. And that's tricky. And this is what we specialize in. And yeah, so I know people like to improvise and but usually when you do that, you get hurt. And in the long run, you might not understand it and see it. In the long run, you're going to get hurt. And I mean, we work very hard to make sure that we try to get it all in the right place. And, we're slowly getting there.
Mm. Yeah, you see all sorts of, very long-term 30-day, 40-day water fasts, people doing them online. So something to kind of think about there. But that last point that you made, so insulin resistance can be protective in an environment where there is a shortage of calories and you're coming into a famine, and then you're going to need to draw down on some of that fat. But then, as you say, if you're in a really big calorie deficit, if I'm hearing you correctly, your body then goes, well, hang on, there's low food availability where we're not getting many calories in here and storage is running out. Let's reduce the amount of energy that we're burning or utilizing as an organism to kind of help preserve some of this energy as a survival kind of tactic. Is that right?
Yeah. And this is, shown by the New England Journal of Medicine, right? So people forget these papers. I'm not, it's entertaining. Like somebody was saying, every 30 or 40 years, people like to repeat the same science. But yeah, these papers are out there, and it's very clear that that's a mistake, right? To not do it, it's a mistake, as 99% people don't do it. So everybody in the world should do at least 1 or 2 cycles a year, just to unlock that insulin resistant state of the fasting mimicking diet. And of course, you don't want to do water only fasting, because most doctors will say, and most experts will say, this is not something that people should do at home. Stop eating. And also because of what I was just saying, the water only fasting may drive you into because it's not clinically tested, and it may drive you into this thrifty mode, even in a few days. Not clear, but some of these studies are not allowed anymore, right? So Ansel Keys back in the 50s and 60s had done these semi-starvation studies in humans, and that was probably the last time in human history those would be allowed. But this, if you read them, they're just remarkable studies of human volunteers that undergo this 4 weeks or more of starvation time. But the biology and the physiology out of that is just incredible.
Yeah, I'm not sure that'll get ethics approval. I'm not sure there would be people putting their hand up to do that. You'd have to pay them a lot of money.
You'd be surprised, You'd be surprised if it was like a study done by a university. There'd be some volunteers for anything. But yeah, I don't think it'll get an ethical approval.
I want to finish here on FMD as a potential adjunct therapy for someone who has cancer. And I know that you had a study that you shared with me looking at mice with breast cancer, where you investigated the effects of FMD. So talk to me, I guess, firstly about the interest in FMD and cancer in the first place, and what your hypothesis was or is.
Yes, my point is that we have that we've ignored in the oncology field is differential property of normal and cancer cells. So I always talk about the analogy of the desert. You take a billion people, you put them in the desert, you give them no water, no shade, and you make them run. And you come back after 2 weeks, and 100% of them will be dead. And if you put them in the same desert and you give them water, you give them shade, and they can let them sit, I would say after a couple of weeks, they're all alive, right? So yeah, a billion people, is it possible that you could change such a few things and a billion people will be either all alive or all dead? Yeah. So the same is true for cancer, or let's say this principle can be applied to cancer. Why? Because the cancer cells cannot stop running. And so if you take away the food, they're in trouble, because they have less food, but they're running. But this is why now the combination with standard of care comes in, right? And that's why we always say, you have this camp of the alternative people and the ones that are obeying some of the FDA and the medical rules, and the ones that refuse them, right? But in fact, it's a combination of both that seems to be very effective. Why? Because now, if you are a cancer cell and you're just giving fasting or fasting-mimicking diet, you can still manage, because you steal from the system, right? You steal from other cells. So you can get the amino acids, you get the sugar, you just deprive the other cells from it. This is why the chemotherapy, the immunotherapy, kinase inhibitors, hormone therapy, et cetera, they're so effective together with the fasting-mimicking diet. So you generate a differential environment, right? So the normal cells, they know exactly what to do. They've been starving for billions of years if you start from the bacterial ancestors. So they know exactly what to do. The cancer cells, just they do all the wrong moves, right? They keep on going, and now they can still keep on going as long as the immunotherapy or the chemotherapy or the radiation comes around. And then it's clear that this is why in mouse model we see, cancer-free survival over and over and over. If you combine the fasting-mimicking diet with the whatever most effective cancer treatment you have for that cancer. Now in people, well, now there's lots of studies for the fasting-mimicking diet in clinical trials, and they look very, very good. So, for example, the 125 patient randomized trial, breast cancer, done in University of Leiden, published a couple of years ago, showed a both clinical and pathological effects. So the patients that did the fasting-mimicking diet together with chemotherapy responded much better. And I think there was a five-fold reduction from 27% to 5% of the non-responders. The portion of patients that did not respond to the chemo. And another fantastic thing about the trial is the dose response. If you look at pathology, so the surgeon takes the cancer from the breast, and then it looks at how many cancer cells are active in that cancer, and then they score it, and it's called Miller-Payne scoring. And there's a dose response. The patient that did all the cycles of chemo with the fasting-mimicking diet were the one that by far had the lowest score, meaning that they had the least active cancer cells within the tumor mass. Right. So it's very nice when you see that dose response. Then another study by Claudio Vernieri at the Italian Cancer Institute, National Cancer Institute. Actually, they did two papers, one on 100 patients and one on 5 patients. But I really like the one on 5 patients because it showed, I think in the title, it had extraordinary survival or extraordinary effects of standard of care together with the fasting-mimicking diet in 5 patients. One had pancreatic cancer. So I really like that because I like the sort of hope factor and saying, hey, maybe if you run out of options, the FMD in this case could make a difference. It may not. We don't know yet, but it could, right? So talk to your oncologist and ask him or her to consider the fasting-mimicking diet together with the standard of care. Because in some cases, in many cases, of course, we follow thousands of patients, but I don't like to talk about that because people then say, oh, yeah, he's talking about a case. He's trying to make a clinical trial out of a case. But we've seen it with many patients. But the clinical trials are starting to show the effects that we've seen with lots of stories. And lots of hospitals are now starting to do it. And I've heard from U.S. hospitals, now they're saying now we do it with every patient. We give them the fasting-mimicking diet. So we'll see, but looks very promising and also very inexpensive, right? People can, even somebody's poor and can only afford to get the old chemotherapy. That certainly water-only fasting, if that's all they got, is something that could make a big difference in their treatment.
In these studies where they're using it alongside traditional treatment for whatever cancers the subjects have in the study, is it being done in the same manner as, say, for example, what you've used in studies for people with type 2 diabetes? Is it still a 5-day thing, or are they doing it every day throughout their treatment, or 10 days? I'm presuming it's not every day because they'd be in too much of a calorie deficit. What's the protocol look like?
No, it's not every day. It's actually shorter. It's 4 days. It's about 600 calories for 4 days. The normal people is 5 days higher calorie. This is 4 days lower calorie. And so this was, now it's been 15 years of arguing with oncologists and physicians and trying to come up with something that we're all happy with. And I think, we're close to there, meaning that, as I mentioned earlier, we don't see loss of lean body mass if it's done correctly. There is a exclusion criteria for, somebody comes in with too much frailty or cachexia or other muscle type degeneration. So then some people are excluded, but I'll say that the majority, or maybe the great majority, can be included or can be brought to a level where they can be included, maybe with some muscle training, etc., and or improve nutrition, and then they start the fasting-mimicking diet. So 4 days combined time with the immunotherapy, with the chemotherapy, with the radiation. So in every case, there is a different design. In the clinical trials, we spent some time with the oncologist to make sure we time everything correctly, so that For example, in the case of immunotherapy, we want to make sure that we don't interfere with the efficacy of the immunotherapy, but at the same time get the maximum effects.
What's it going to take to kind of build on your confidence as to how effective FMD is during cancer treatment, and potentially to be, say, included in cancer guidelines and to have more oncologists? I know you mentioned that there are plenty that are using it, but to kind of get the word out there to more and more oncologists, what further research is required?
Yeah, first of all, I think the patient is number one, meaning that the patient should, if the oncologist said the treatment is not working, I don't see it. I don't have much hope. I think it's good to go to oncologist and say, wait, can we try the fasting-mimicking diet together with whatever it is that you recommend? And then, of course, like for everything, it takes a long time, and that's unfortunate because I think I always say the science goes very fast, and the FDA goes very slow. And there are good reasons for the FDA to go very slow because they have to make sure they filter out a lot of ideas that are not true, right? So with COVID, we heard lots of stories right at the beginning of what was making patients healthier, and then the great majority of it was not true, right? So then oncologists know this, and they basically say most of the stories we heard were just stories, and eventually did not make the cancer patient live longer. So I think that it's going to take a long time, and but there's a lot of trials, and I think that as the success stories like the one I just mentioned come out, and the papers in animals increase, then I think there's going to be many hundreds. Like I don't know if this is going to have the potential of immunotherapy. But let's say that it did, then eventually there's going to be hundreds of hospitals that are going to start doing clinical trials on immunotherapy, and this is what happened. So immunotherapy was first discovered in the mid-90s, right? It took a long time for the explosion. Probably around 10 years ago, you started seeing the explosion on the clinical trials on immunotherapy, and then the success stories in various cancers, right? So then from the 95 discovery of immunotherapy to the now, it's 27 years. So 27 years to get to where immunotherapy is a central part of cancer treatment. And that's the way it is. And, and part of it is because you have so many cancers and so many therapies. You have to eventually check everything with everything, right? So immunotherapy in together with some other drug for the treatment of this stage of this cancer, right? So there are really hundreds of combinations, and that's why you need hundreds, or if not thousands of studies.
What do you think about the ketogenic diet? Because that's another kind of diet that gets thrown out there as a possible or possibly being beneficial for folks with cancer, moving to a kind of low glucose diet. You hear people saying that glucose feeds cancer cells. And I know that there's the FMD is low in kind of high glycemic carbohydrates or refined sugars. So is there some overlap in the sort of thinking behind these two different ways of eating?
Yes and no. Obviously, the fasting mimicking diet and cancer, it's a very theoretical, it started with yeast and very fundamental ideas, right? So and that's why the starvation part is the most important part of all, right? So you want to create a differential property. In the ketogenic diet, you start with the wrong food, right? Because you're giving high calories, right? You're giving normal calories. And, and cancers, usually many cancers, and we know this, love, the ketogenic diet. And many cancers hate the ketogenic diet. And many cancers could hate the ketogenic diet first and then start loving it because one cell is mutated and that takes over, right? So So I think that I'm very supportive of lots of studies on the ketogenic diet, and we're now combining it with the FMD and see, for certain cancer, can we get even a better effect? But I think that in general, the FMD, I think, is going to stand because of the extreme calorie restriction. Enough days, so 4 or 5 days, enough days to try to kill as many cancer cells as possible, but not enough time to disrupt the normal feeding of the patient. So there is a lot of pluses, but the ketogenic diet does allow you to eat all the time. So it's interesting, and that's what we're testing now. And I think other people are testing. And I know that some of the clinical trials with the ketogenic diet also had the fasting or a fasting-mimicking diet with it, even though they were described as ketogenic diet trials. So I think that we need to look at it, and we need to certainly find out which cancers love high fat and high protein. Then distinguish between high protein ketogenic and low protein. I would say high protein ketogenic should be out, and it should be only the low protein ketogenic. And you got both right out there in the clinical setting. You got the high protein ketogenic and the low protein ketogenic. So there's no reason to go high protein. I've seen no evidence whatsoever, but the low protein ketogenic could be promising for certain cancers. So one thing we're testing now is like normal diet, ketogenic diet, the fasting-mimicking diet, in cycles. And sometimes, so then sometimes you exit the fasting-mimicking diet and you go into a low-protein ketogenic diet. And we're getting, not surprisingly, mixed results, right? Some cases is working very well. In some cases, not working at all, and maybe even making things worse, So, and the good thing about the fasting-mimicking diet, now maybe 20 laboratories, so far we haven't seen a single negative. It's coming, right? It's coming. I know it's coming, but it's interesting, right? After 15 years, we haven't seen a single negative study on the fasting and fasting-mimicking diet in cancer. So that's very good news, right? That it's going to be out there. We're going to see something growing more, some cancer doing worse during fasting. But thus far, nobody is published that. So very, very, very promising. At least that if it does make something worse, it's fairly rare.
Mm-hmm. Definitely, Valter. This has been extremely interesting. I really appreciate you taking the time to come back and speak with us again. Was there anything related to the topics that we spoke about today that you wanted to kind of add before we wrap this up that you perhaps didn't get a chance to touch on, or do you think we covered it all?
One thing, the paper we just published, so we published a paper on immunotherapy and cancer last week, but we also published a paper on Alzheimer, which included the first 30 patients, University of Perugia, University of Genova. And I just wanted to say, it was a mouse paper, so very powerful effects of the fasting-mimicking diet in mice. But also interesting that we could get the patients, the Alzheimer patient, to do it for a year. And there was a dropout, but it was a very similar dropout between the control diet and the fasting-mimicking diet. So that's an interesting. It's just a promise there because in humans we have no data on cognition yet. But I thought it was very important to start thinking about, in addition, so many drugs have been tested for Alzheimer and they're all failed. And so now I think it's interesting to think about combining it with the fasting-mimicking diet as we're doing for cancer. So we're just encouraging anybody that wants to do trials in that division or anybody that wants to talk to their neurologist about, also introducing a nutritional treatment or potential treatment for Alzheimer's. I just wanted to mention that.
Absolutely. Fascinating. Well, thanks again. And as always welcome to come back on and speak with us whenever you want. Perhaps if you have any new studies coming out that you'd like to share with us, that would be great.
Sounds good. Thanks a lot.
Thanks, Valter. Thank you for joining me for this episode and your interest in science-based conversation. I hope you enjoyed it and found the information covered interesting and instructive. If you did and you'd like to show your support for the show, please subscribe to our YouTube channel where you can stay up to date with new episodes and watch them in video format. Yes, the full-length videos. Please also consider subscribing to the show on the Spotify and/or Apple Podcast app, wherever you enjoy listening to podcasts. You can also leave a review on Apple or Spotify. Again, a great way to support the show and make our content more discoverable for others to enjoy and learn from. If you have any comments about the episodes, suggestions for future episodes, including guests you'd like to see on the show, or questions that you'd like to have answered, please leave those in the comments section on YouTube. I myself and my team will take note of these comments when planning future episodes. Finally, the best way to support the show and receive discounts on products we love is by checking out our sponsors at theproof.com/friends. Enjoy your week, stay well, and I look forward to catching you in the next episode.